Namaste students, Khushro Betia. I am JP and Dr. Vidyapeeth, Assistant Professor Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the cytoskeleton. The microfilaments are involved in the movement of all the mobile cells, including non-muscular type, and drug that disrupt the F-acting organization. Effect of the activity of these cells all actin comprised of 2% of the total amount of the protein in hepatocytes and 10% in the fibroblast or 15% in amoebias, up to 50 to 80% in activated platelets. So there are a number of different types of actin which slightly different in structure and function, which means that an alpha actin is found exclusive in a muscles fiber while a beta and gamma are found in other cells. In addition to that, a high turnover rate of the majority of which in the outside are movement cells, and this means that microfilaments found in the cells other than the muscle cells are present in three forms, that is microfilament network, microfilament bundles, and periodic active ring. So microfilament networks in animal cells the commonly have a cell complex or which animal cells have here cells which have a cortex under the cell membrane that contain a large number of filaments with and the presence of organelles. This network are connected with numerous receptors and rely on a signal to the outside cells. The microfilaments bundles are extensively here located at the network in association with the contractile proteins and as non-muscular myosin or a periodically actin rings which may be spaced as actin ring found in the axons or not in dendrites or the actin ring together with the spectrin on forming a link of the neighboring actin and form a cohesive cytoskeletons which supports with the axon membrane. Periodically may be regulated by a sodium ion channel in axon. In the yeast, it can be seen the actin cytoskeleton, which process a key endocytosis or a cytokinesis or determining the cell polarity on morphogenesis in yeast. So, in addition to which, the actin in this process have 20 to 30 associated proteins, which have high degree of evolutionary uh, conservation along with the many signal molecules. So, yeast contain three main elements associated with actin are patches, cables, and rings. So here, despite not being a long such a dynamic equilibrium during the continual polymerization and depolymerization of this protein, in plant genome, it is revealed that the existence of the protein isobariant within the actin family of Arabidopsis thaliana, a dipod plant, when used as a working model here, model organisms. So there are 10 types of actin, that is 9 types of alpha tubulin and 6 beta tubulin and 6 profilin and a dozens of myosins. The diversity which explains evolutionary necessity for the processing variants in which may be slightly a temporal or sprital expressions. Majority of the plant have the cell wall that defines their morphology or the movements of the microfilaments that generate the sufficient force to achieve a number of cellular activities such as the cytoplasmic current or the generation of the microfilaments in the myosin or the actin which involves in the movement of the organelles in the cellular morphogenesis or cell divisions or elongation and differentiation of a cell. The notable protein associated with the actin cytoskeleton in plants are the valine, which belongs to the family of the gelsolin, and which are able to cut the microfilaments and bind to an active monomer in the presence of the calcium cation fibrin, which is able to recognize and unite the active monomer. So, the formin which are able to act as a, F-actin or a polymerization nucleating agents or a myosin, a typical molecular motor here specific to eukaryotes, which is found in the Arabidopsis, which is coded as a 17 gene in two distinct classes. Okay. So the Golgi apparatus as well as composed of xyloglucans in the cell wall, and which forms the emergence of the actin cell structures and associated with the membrane of the microfibrils, and which reaction involving the organism's reaction to the stress. Nuclear actins, which are first noticed by Clark. Transportation of actin through a nuclear membrane where in the cytoplasm operant binding ADP actin monomers, which forms actively imported into a nucleus, or a high concentration of ATP in the nucleus when compared with the promotes the ADP to ATP exchanging actin coupling complex. This speaks the strength of a binding and these two proteins. So coupling act as a here complex finally when associated with that of the nuclear limb kinase. Okay. So here the organization of a nuclear actin exists in the monomer but not in the form of a dimeric oligomer or a short polymers. So the nuclear actin or the organization or which varies in a different cell type or having a high level of comparison when it compared with the somatic cells. The nuclear actin function as the architecture of the nucleus, transcription, regulation of a gene activity, translocation activity of a chromosome fragment, or a integration of different cellular compartment, or a immense or immune response, or a muscle contraction, the outline of the muscle's contraction. So here the actinomyofibril makeup may be the for the cytoplasmic materials forming a motor protein in a myosin. The active role in a muscle's contraction, depolarizing of the sarcolemma in the transmission of the action potential in a T-tubulin, or opening of the sarcoplasm reticulum in the calcium channel, or increase in the cytosolic calcium concentration interacting with the ions and forming calcium captures the sarcoplasm, causing few conformational changes which inhibit the actin myosin interaction or other biological interaction of cytokinin or apoptosis or cellular adhesion and development in or gene expression. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving session. Thank you.